basically to to discuss our class, what's going on so far and how we are going to proceed in the future. Okay. And also, I need to go through two important things here. The first will be the lab. I need to discuss with you more about the lab. First, as you know, the lab is basically virtual and you can do anything with your practical, unfortunately, which is considered very tough for biology and microbiology, but we are trying to do our best to make things kind of easy for students so they can understand the experiments, the objectives, how to interpret the results and stuff like that. Because basically in your practical, which is coming in 2 weeks, practical one, you are going to see something similar exactly. So you're going to see an experiment, like a bacterial culture in a media, and it will ask, if it is the color development, for example, say for example, motility, will give you two test tubes, one of them showing motility and the other one showing non-motile bacteria. So it will ask you which one of those represent motility. It will ask what kind of indicator we use in this experiment. So same deal like what you are doing for the assignments. So assignments is a very, very good practice for you. If you do your assignments, that will make the lab practical section or part very easy for you. So I have noticed that some students, they didn't return their lab assignments as frequently. Some are very, very dedicated and they put everything on time, but still some students are missing those labs. Don't say, okay, this lab is 4 points, this lab is 6 points, so it doesn't matter if I do it. Will, don't look at the points now, but look at how to understand the lab, how that lab will make you ready for your practical. So this is very, very critical. So I'm emphasizing on this. This is why sometimes I extend the due date for lab, to return lab assignments, just to give extra chance for students to do that. But unfortunately, I'm not going to do this again because I have other things going and it will be hard for me to go back and forth, to extend due date and stuff like that. That will be very time consuming for me. So if you don't mind, I will keep the lab assignments open and I will be waiting for those who didn't participate or who didn't do their lab assignments to return their work. I will give it a couple of days and then I will start putting, for those who didn't email me the assignments, unfortunately, I'll start putting some zeros on this. Okay. So I sent you few days ago a very important lab, which is Staphylococcus identification. And there are 7 species for Staphylococcus. And I assigned for each 2 or 3 students one, but you need to work individually. It doesn't mean that if you have if you see isolate one, for example, 4 students, that you need to work together. No, it is this isolate is assigned for those names, and each one will work individually to return the lab the identification sheet for staff. Also, please include a 2-page essay about the species that you was able to identify, in terms of if it is a pathogenic, what kind of disease this particular species cause, what about virulence factors, and how this particular species can be treated, what kind of antibiotic can be used to treat a disease caused by that particular isolate. Okay, so far, if you have any question, please ask me. I'm still doing the talking about the lab section. Hello.
I'm good.
Yeah, if you have any question, ask me. Anybody else has any question for me at this point? Okay, so now I propose for you to meet like 1 day a week for a lab and 1 day a week for lecture. Okay, so for lab, for example, we need to meet and discuss some of the lab experiments or some of the lab essays that it was kind of hard or kind of confusing for you by looking at the PowerPoint presentation. And we can discuss those, like if there's any specific question or any specific uh, subject that we need to discuss, we can go through this in our lab meeting weekly. So I prefer to do this kind of lab meeting like on Friday. I will send you like a Zoom link for a meeting. And again, I will also record it for those who cannot attend. It will make it easy and more efficient if I receive emails from you with some question about a specific essay or a specific lab. That will make it very time efficient. You know, we don't need to go through everything. So if it is not really necessary, if it is easy, straightforward, there is no need to do this. But my point is, those labs or assignments or questions or essays that you feel kind of hard to understand or to make sense out of it, you just drop me a line, an email or something, and tell, okay, I would like to discuss this in our next meeting. And this way I will be basically preparing those so we can go in details about these questions. You have any, I'm open for any suggestion. Hello?
That sounds good to me.
Any suggestion? Any question for me at this point? Okay, so generally speaking, you have any question for me for the lab section, lab part?
I don't, not right now.
Okay.
I'm good as well.
Okay, great. Okay, so now let's shift to the lecture part. Okay, for the lecture also, I'd like to meet with you like once a week, again, via meeting, Zoom meeting. Again, I also like to do the same with lab. So say we have 2 chapters covered, you know, this week. So what I'm doing is basically I send you the PowerPoint presentation ahead of time so you can go and read those PowerPoint presentations. The PowerPoint presentations basically summarizing the chapters. Okay, so it's the points, you know, and if for any reason you feel like there's some information which is not really covered deep and you'd like to have more information, so you still have the textbook as the backup for this part. So I'm sending the PowerPoint presentation and I will be waiting for also like comments or questions to be sent to me about specific slide or a specific subject or a specific part of the chapter that was kind of hard to understand in your own. You email me this, so when we have our meeting on Friday also, so we're going to, sorry, that will be for Monday. So the lab, we're going to meet for the lab on Fridays and for the lectures on Mondays. So you send me like those like questions you have so I can like discuss more details about it. Also for the exam or test, I'd like also to do like a review session before the lab. and it will be also more efficient if you email me again, like something that you don't understand. You can get a question you have. So when we have our like review session, I can go through those deeply and thoroughly. As far as I know, till now you did very great job in test number 1. As a matter of fact, I can see roughly that I have a maximum student get a maximum of 96 out of 100 and a minimum of 75. And the average for the test was 88, which is great. Very, very good. I will basically give you also some extra credit assignments to help you if you feel like you didn't do good in a specific exam. Okay, so I'm going to follow up with like an extra credit essay or question or something about that particular part that was covered. I will tell you exactly what you need to focus on. So that will help you like jump and move your grade a little bit higher. So that is what also I'm going to do. So again, I'm listening for any suggestion you have for me.
Okay, I have 2 questions.
Yes, go ahead.
Do you know what time on Monday you'll have the meetings for the lecture?
That's a very good point. So again, unfortunately, I don't know exactly, your time, because every student has his own or her own schedule.
Right.
So I don't know. I think it would be a good idea if you just email me like day or night, and based on the number that I'm going to receive, I will decide, we'll schedule for that particular time. But remember, if you can't attend for some reason, you still will have the recording for the meeting. So the recording, when I do recording, it is big, huge file. So I'm going to post it on a YouTube channel for you. I can post it on eCampus. Those kind of files are heavy. So that was one question. What else? You said they have 2 questions for me, right?
And then regarding the extra credit,
Yes.
I know there was a section on the test and I wasn't really sure what to do there, where it said there was 5 points extra credit.
No, that was nothing. That when I say you earned, it means that
was
you
the
earned
thing.
already. You don't need to do anything about it.
Okay.
Some students say thank you very much. Some say very good words. Some didn't even say a word, which is okay. So don't worry about it. Yeah.
Okay, so then I know there's something in Blackboard right now that's due tomorrow. Is that additional extra credit for this first test?
Okay, let me see here what I have. It's probably I put just a title, but I didn't do it yet. No, I will give you ahead of time for sure. When I do something like that, I don't, I don't decide to put it today and then I say it is due tomorrow. No, I never do this. So I will give you an I will give you definitely, like I will announce for it. I will give an extended time. I will give you plenty of information. So you will have an idea what is going to happen, what you need to do, when, all this information. So whatever you see now, maybe it's still unavailable because I just put I didn't put any information yet on it. I just kept like a spot for it on the gradebook. Yeah.
It says extra credit to be added to test 1, and when I click on it, it looks like it's a little quiz. It says there's 15 minutes.
But I didn't add any question yet, right?
I don't know. I haven't tried to test yet. I was waiting until after this.
Yeah, meeting. Because it is not open yet. Yeah. Let's see here exactly what I have.
It says it's due tomorrow, so I was.
Yeah, let me do this and see exactly what we have here.
I didn't have to do it yet because I was going to wait till after this meeting to ask.
Yeah, let me see what we have. Okay, assignment quizzes. So you have, it says extra credit to be added to test 1 if I open this one here, yeah, as I mentioned, I just didn't start it. I just put like a title for it, but I didn't finish it yet. So when I finish, when I have it ready, I will put an announcement, and you will see it in your grid book, you know, about the start, when it is going to be available, and when it is going to be due. So you don't need to worry about it right now.
Okay, thank you.
Yeah.
I have a question.
Yes.
I joined a little late to this meeting, but so if it's already been asked, I'm sorry. One of my questions is about the quizzes. Like I know that you can see which ones you got wrong, but one time it like kicked me off, so I never got to see which ones I got wrong. And then I would have looked up the answers had I known which ones to look up. But I was just wondering if you have an answer sheet, something after we.
I heard the first part of your speech, but then the last two sentences I didn't get because it was cutting out. So what was the question again? For the
Oh,
quiz?
just that if you have available the answers to the quizzes that we can see after we've taken them. Yeah.
Yes, definitely. This is what I do. Basically, I have the option to show students, you know, their answer and the right answer after the due date. So when I put the quiz, I give the option, you know, so you have you can access your quiz, and you know exactly, you know, what did you miss. And as I mentioned earlier, we're going to meet like every Monday for like discussion or for lectures and stuff like that. It would be a good idea also if we can go through the quiz or quizzes and see exactly, you know, how students do, and you know what is the right answer should be. But definitely, when I put a quiz like this in Blackboard, I give the option that students can see the answer and the right answer after the due date. But I will
Okay.
also make sure definitely that that you know that will be for every test or every quiz. Yes.
Okay, and I had another question about the lab practical.
Yes.
Is it on a specific day that week that it's on our syllabus?
Yes. It is basically the practical will be, I believe, not in 2 weeks. So not next week, at the week after, I believe.
Is it? Do you know? Is it on a Thursday or what day is it on?
This is basically the whole department need to decide about the specific day, because they need to give all the sections need to do it at the same time.
Okay.
But that will, I already have the week posted for the practical. Yes.
Right. Okay. Okay.
Yeah, but again, I will give you like plenty of time. I will tell you exactly when. And when I open it, it is not going to be like open for a few hours. It will be open like maybe a couple of days. So you can schedule your time based on this. Yes.
Also, I have a quick question. So for the practical, I know that I took AMP, and like how that will be different compared to this class. Like, would it be like labels? Like, what is the best way to study that? Like, will it be just to, like, study the previous labs, or what? Or will you give us materials?
It will be exactly like the lab assignments that you are doing right now. So it will be, some of them will be like just fill in the blank and put the right word, like what kind of, say we're doing like endospore staining, for example. And then what is the name of the primary stain? Say, so you need to write this down, malachite green. Saying Gram staining, what is the name of the counter stain? Okay, it's safranine. Maybe I'll give you two tubes, tube A and tube B, one of them showing motility and the other one showing non-motile bacteria. And I will ask you which one of those tubes showing motility. So you need to say tube A. It will be like that, you know. It is not too much.
That makes sense.
Yeah, not too much writing at all. It will be multiple choice, maybe true or false. fill in the blank with very little words, you know.
Oh, okay.
Yeah,
okay.
I will not ask you how you perform gram staining or how you perform capsule staining, or you know, I will not ask you to do or to write down a complete experiment for me, you know,
Oh.
for this. Yeah, no, that is not going to happen.
Awesome. Also, I came a bit late. Did you talk about the group project that's coming, like the lab assignment? I know that you assign us each the Staffla ID for each group.
Yeah, no, this is, yeah, that's a good point. I just mentioned this earlier. I said, when I put, you know, like an isolate for you to identify and put names, like 3 names or 4 names, it doesn't mean that you need to work together. No, that strain or that isolate was assigned for those 3 names. Okay, so each one will work individually. Each student, you will work individually in your own. You don't need to communicate with anybody. Okay, you are going to do it in your own and email it to me in your own.
Okay.
Yeah,
Okay.
it is not a group, like kind of a group assignment that you need to meet with those names and discuss it. And no, that was the plan if it is like face-to-face course, like what we used to do in the lab, real lab. But here we are doing virtual lab, so there is no need for students to get together and discuss and you know, send like one report for all group. No.
Okay. That makes sense.
Yeah.
Thank you.
Yeah, no problem. And like in end of this week, I will send again for each student, I will give you a name of a bacterium that you need to have a presentation or a PowerPoint presentation. For the PowerPoint presentation, there is no need to for the student to present it. So we don't need from you to do like a Zoom meeting and present what you have, but you are going to send me the PowerPoint presentation. I will send you exactly, you know, what you need to include in your PowerPoint presentation. You are going to do it, email it to me, and then I will post it for the group so they can look at that particular organism, what kind of information you have. So end of this week, I will put together this assignment for you. And this is for the 50 points presentation. If you look at your syllabus, you will see there is a section which worth 50 points. And that is the presentation that you are going to do. Although there is no presentation, it is basically you are going to put together a PowerPoint and email it to me. You don't need to present your findings. Did you get that one?
Yes, that makes sense. Thank
Okay, great.
I have a question.
Yes.
So on the staff ID that's due on the 6th of July, we are to turn in a PowerPoint presentation and the 2-page report.
No. For the staff ID that I sent you, remember I sent you a PowerPoint presentation about your isolate, and I sent you like an attachment, like an identification sheet. You know, that 1-page sheet, gram positive or gram negative, what is, you know, shape, you know, if it grows like in mannitol, salt media, all this kind of thing. You need to fill those for me. Okay, and based on this information, you will tell me that my species is whatever, Staphylococcus aureus, for example. This is one part. The second part, you are going to write down for me like an essay, like a 2-page about Staphylococcus aureus. You know, the organism, you know, where you find these, some characteristic feature of the organism, if there is any virulence factors associated with that particular species, what kind of disease and how that disease can be treated, what kind of antibiotics are most effective, all these kind of things. Just 2-page attached to that identification sheet that I sent you. You don't need to do any presentation or anything like that.
Okay, so those are 2 separate like assignments.
Yes. Remember, there is something in the lab which is staff identification, and there is something called PowerPoint presentation, and that is for the lecture part. For that one, I will give you the organism. I say, okay, go ahead and talk about Bacillus cereus, or go ahead and talk about Clostridium difficile, you know, So you need to do a PowerPoint presentation. I will give you the guidelines, how, what you need to include in your presentation. Then you don't need to present it for the students. You don't need to do that. You just email me the presentation, like, my presentation, and I will post, I put it in the eCampus for students to access it.
Okay, so the assignment that's due on July 6th is the staff ID 2-page report.
Yes, that's
Okay.
it.
And
Yes.
the PowerPoint, we just have yet to like get any information over.
Yes. End of this week, I will send you more detailed information about it.
Okay, that makes sense. Thank you so much.
No problem. Okay, do you have any question for me? So let me go through exam one with you. Let me share my screen, because I'd like to show you, as I mentioned, you really did, everybody did very good job in that particular exam. We have an average of 88%, which is not bad at all for the first exam. And that is the first exam. I believe it's kind of a hard one because it has lots of information, especially some background information about the subject. Most difficult chapter I can see was chapter 14. But I didn't give too much question for that one. So can you see my screen?
Yes.
Okay, so this
Yeah.
is some statistical analysis for the test, and there are some questions that was kind of not confusing, but was kind of hard, and I had different kind of response, for those. So I need to emphasize on this particular question and tell you exactly the right answer and why this is considered to be the right answer. So one of them was basically question number 7. If you look here, I have almost 90% of the students say it is false, okay? But it was right, and I will tell you why it is the right answer, or true answer. So heme prosthetic groups are involved in electron transfer with quinones, okay? So I have here another, I need to show you this PowerPoint presentation to tell you why it is true, okay? Okay, can you see this screen?
Yes.
Okay.
Yes.
Yeah, so this is basically, it talk about the electron transport chain in the mitochondrial membrane. And as you know, there are several protein complex involved in the process of electron transport or transfer. We name those complex 1, complex 2, complex 3, and complex 4. So if you look carefully here, you will see that quinone is basically sitting in the membrane because it is hydrophobic. So it is basically sitting in the membrane, and it is basically, its main job is to take electrons from complex 1, complex 2, and move it to the complex 3. So if you look here, you look at complex 3, what you can see, complex 3, which is cytochrome, it has a iron prosthetic group sitting there. So it is basically, yes, there is a correlation between the electron transfer from the iron and from the quinone based on this information. Okay. So that was why it is a true statement. So quinone is basically taking electrons from complex 1, complex 2, and move it to complex 3. And what is involved in the complex 3 is the cytochrome, which has a prosthetic group in it. Okay, so that was one of the questions that I wanted to go through. And let me go back to the test. And there's another one, question number 15. So if you look at question number 15, it says a direct microscopic counting of cells is considered to be like an accurate method for estimating the number of viable cells in a sample. It is a false statement. Why it is false statement? Because when you take a sample from a culture and you look at under the microscope, you will see cells, you will be able to count, but you cannot differentiate between dead and living, viable and dead cells. You cannot. By just simply looking under the microscope, it will be very tough or very hard for you to distinguish between viable and dead. Okay, so you can't do counting for viable based on microscopic examination. Okay, so that shouldn't be, that should be a false statement, not a true statement. Again, I have 76% of the students say it's true. Only 23 says false, and this is the right answer. Okay, you have any question for me? Any comments? Question number 19. It says here, autoclaving and pasteurization are two processes that are both very effective for sterilization. Word sterilization here, meaning eradication and eliminating of 100% of living organisms. Pasteurization is not eliminating 100%. Okay, autoclaving do, but pasteurization no. Okay, and if you look at the milk that you are drinking, for example, they have a due date. So what is the due date all about? Because milk during the process of packaging, they do pasteurization to eliminate a specific number of microorganisms. It is not complete elimination. And they give you like a due date telling you that, you know, the due date say in 2 weeks, meaning in those 2 weeks, the number of the microorganism will be very limited. It will not going to do any spoilage. And then starting 2 weeks after the due date, you will start see spoilage of the milk. Why? Because there's still some microorganism living there and they are increasing in numbers to the extent that they're going to spoil the milk. Okay, so autoclaving is a very good sterilizing method. Pasteurization is no. Okay, just for your information. And the last one also was question number 24. A pure culture, when you identify a word pure culture, it is basically a culture which is made of a population of identical cells. So if I have a pure culture of E. coli, it means that I have 100% E. coli cells. Okay, if I say it's a contamination, if in addition to the E. coli, I have Bacillus subtilis, that will be a contaminated culture. I can't say a pure culture is sterilized. I can't say this. I can't say that a pure culture is made of defined chemical media. There is no relation or correlation between a pure culture and the kind of media that is made of. Okay, so the way to identify pure culture is basically population of identical cells. Okay. Other than that, you did very good in the rest. That was the main question that I have seen that was kind of confusing for students. The last one, it was talking about the structure of flagella, bacterial flagellum, and some students was kind of confused a little bit because the space left was just like for one thing only. This is the way when I put that question, I fill in the blank and I put it in the blackboard. Unfortunately, that was the only option I have, that I have only one box to have all, you know, the answer. So just this is out of my hand. Forgive me if it was kind of confusing for some of you. But again, majority did a great job in this particular question here. Can't see like okay. So yeah. Do you have any question for this exam? Any particular one that you would like to go through? Was kind of confusing for you? Hello? Hello?
I'm good on my end.
Okay, so good. So this is basically what I have for you today. So do me a favor if you don't mind. And for those chapters, again, email me if you have any question, anything that is not, you can't understand or you can't comprehend very well. And then in our meeting each Monday, we are going to discuss those. Also, if there was any kind of quiz that was given during the week, also we're going to discuss that particular quiz or the test. I'd like also to do like a review session for the exam before the exams. And the same deal for the lab. Also email me if there's any experiment or any assignment that you can't understand, so we can discuss this in more details.
Okay, thank you.
Thank you very much. Any question for me before we finish?
Yes, to clarify, so for the quiz tomorrow,
No quiz tomorrow. I didn't finish that yet.
for the
There is-
extra credit quiz,
Yeah, nothing
worry
yet.
about
It is
it?
not
Okay.
yet. It is not yet available. I will give you information, more detailed information when I have it ready.
Okay.
Yeah, I will put it in the announcements. Okay, and you will see it clearly, you know, the due date and stuff like that. Yeah, okay. And by the way, you are doing great job. You are doing very, very well. And it looks like you're going to be the same like my spring class. I have my spring class. I have like 65, 70% end with an A. And the rest was B. So I got like fired like by the department. How come, you know, all those students got this A? It's too much A's, you know. They are very, very good in terms of like returning things in on time. You know, they are very, very patient, you know, to the end of the class, and they did great job. And I believe you are going to be the same, based on my first, you know, test or exam. I can see that when you say about an average of 88%, very close to 90, it is like an A. That's great. That is great.
I have quick questions. So for the lab, you will send more information this week, right? That's what you said for the report.
For the which report? Remember, we have two things.
Right.
We have the staff ID. Already the information was sent to you, and we have the presentation, and this is a 50 points presentation, and I'm going to send more information toward the end of this week. I will give you the name of the organism or the bacterium that you're going to work with and to present.
Okay, that makes sense.
Yeah. Okay.
Okay, thank you so much.
Thank you very, very much. And I'm going to post this. I put a link in YouTube. I will put it in YouTube and send the link for the class. Okay, you guys enjoy the rest of the day and have fun.
Okay, thank you.
Don't work too much. Okay.
Have a good day.
You too. Bye-bye. Bye-bye.